Just wanted to really begin by congratulating everyone on the Total Body PET and the launch. And I think everyone is equal, but I would like to single out a few people. So Steve Meikle, who proposed the idea at the beginning and has been the advocate all the way through. I remember discussions with the executive of the Brain and Mind Centre and then the incorporation with DVCR, and I think there was strong support for the application. Obviously, Fernando from Sydney Imaging, and then obviously the involvement of Simon Ringer and your successful bid. Then the question was, where is the best place to house the Total Body PET? And I think you can see today that that's an excellent decision to have it at North Shore and incorporate it with clinical and research. And I think that's a great model going forward. So this is our Brain and Mind Centre group, and this was our annual symposium. And I think we've got a focus on brain development, mental health, and then healthy brain aging, which brings in all of the neurological conditions. And I see many potential approaches in Total Body PET through these clinical audiences. So this is actually a slide that I use for medical student teaching, and I'm sure it's very interesting, the answers that you get back. But I'm sure everyone here knows that the most important organ is the brain. And all of the other fields are great subspecialties. So we just heard about cardiology, the massive muscle at the end of the vagus nerve, neuromuscular medicine, respiratory medicine, the diaphragm connected and controlled by the brainstem, obviously gastroenterology, all of the plexuses under control, direct control of the brain. So far, no one has been able to transplant a brain. And I think that's been the hierarchy, the importance of the brain. But I think the PET now, Total Body PET, is going to give us some ways to get back into understanding brain function and hopefully to be able to unlock a lot of the unknowns in particularly neurodegenerative disease. So patterns of disease spread, progression, and ultimately, hopefully unlocking new targets for therapies so that we can improve outcomes for patients with these horrible conditions. And I suppose from a clinical perspective, we're down at the bottom right-hand corner there. We're at the diagnostic level. So we're basically here. We see patients when they present with symptoms. But what we don't really know is, when did this condition begin? And we know that each of the various neurodegenerative conditions are associated with specific proteins, whether it's amyloid with Alzheimer's disease, alpha-synuclein with Parkinson's disease, TDP-43 with motor neuron disease. We've got no idea when that process starts to lay down. And it's probably linked to actually the process of brain development. And the brains, when they're put together in the first trimester, are excitable, hyper-excitable, and then gradually with development, they become inhibitory, and then we're born, and then this process triggers off. And I think that we have to really be able to get to the bottom of these processes to come up with effective therapies. And our approach to date has been very much the model at the top is Jean-Martin Charcot, who was working at Salpêtrière in Paris, and he described multiple sclerosis, ALS, FTD in the 1860s, and he used a pathological approach. That was the technology of the day. And these are some more recent studies driven particularly through Glenda Halliday, and using the brains from brain donors at our various clinics at the Brain and Mind Centre. And we've been able to look at brains and work out patterns of disease spread. So these proteins don't just come up everywhere. They begin in certain areas of the brain and then spread through the brain. And that in turn leads to the generation of various symptoms that the patients present with. And so again, this is work that has been driven through Glenda, showing that you get focal involvement, for instance, in the motor cortex, then spread into the frontal regions. And that's the executive function of the brain that determines everything we do, and ultimately does spread everywhere, involves the brainstem, and of course, patients die from respiratory failure. But this is a pathological approach, and we can't do it in a time-sequenced pathway as yet. We haven't been able to do that, and that's what I think one of the key opportunities with the Total PET moving forward is. And this was some research undertaken by Ineda Miyoshi, who's recently been appointed as the first chair of occupational therapy in the United Kingdom. When she was working with us, she talked to carers, and they identified that for 4 or 5 years before their partners developed these conditions, they'd noticed behavioural changes. They'd make suggestive remarks, use catchphrases, they're impulsive, decline in usual interests, and apathy. And these symptoms are there for at least 4 to 5 years before patients present to their doctors ultimately with a neurodegenerative condition. And linked to that then, how do we know when someone is going to present with neurodegeneration? Well, one of the ways that we can look into this is there are families who carry genetic mutations that are linked to disease, and we can follow them in a sequential fashion. And that's what we're doing at the moment. And what we see is when we start treating them now, because there are therapies available, so antisense oligonucleotides, we can give them genetic therapies once they start manifesting these conditions, for some conditions at the moment. But what we're inevitably going to see is a whole range of therapies, but we don't know when to treat the patient. So individuals may have a genetic mutation and may die with a genetic mutation without developing the disease. One way is obviously to look at a change in neurofilament levels. The next thing is, can we try and link these changes in blood with brain function to better identify when to start treating patients? And the importance of that is that for all of our patients now who we see clinically, we undertake genetic testing for these conditions to basically work out, are they amenable to genetic therapies? So linked then to neurodegeneration is involvement across all of the systems. And I think this has been only relatively recently started to be understood. And that relates to alterations in bowel behavior, in lipid behavior, insulin resistance. Obviously, we knew about sleep disruption. And there's a whole multi-system process that is linked to neurodegeneration. And in fact, if you bring all of the neurodegenerative diseases together, one common theme they have is alterations in insulin resistance and metabolism, whether it's weight loss related to Parkinson's disease, hypercholesterolemia and weight loss in motor neuron disease, and just alterations in behavior linked to eating. And again, we follow this from a clinical perspective. We notice that once the body mass index starts to go up, that's usually an indicator of a specific type of alteration in eating behavior, which is underlying, which is caused by the neurodegenerative disease. And particularly frontotemporal dementia is that type of a condition. So we know that hypermetabolism, alterations in lipid resistance, and these are ways to better understand neurodegeneration, but they also have prognostic factors. And if we start to treat them, the outcomes are better. So even if you have someone who's got a genetic condition like TDP43 frontotemporal dementia, if we treat all of their vascular risk factors, the outcomes are much better from a clinical perspective. Now, linked to obviously this alteration in eating behavior, we've been quite interested to see what parts of the brain have been driving this. This was a study undertaken with Rebecca Ahmed and Asa Peterson in Lund in Sweden. And we sent the brain samples over from our Brain and Mind Center clinic, looking at the alterations in neurons, selective neurons that express orexin and oxytocin. And sure enough, they were reduced in the hypothalamus. So it's this alteration in brain structure and function that's driving these changes. And that's where, again, I think that we can start now to try and put the changes that we know that are occurring clinically with the brain imaging, but then the full body PET approach to better understand and link that then to the spread of the disease and ultimately target the neurodegeneration. So to put some of that into perspective, this is a common presentation. People may present with the loss of a muscle here, first dorsal interossei, associated with loss of the adductor pollicis brevis. So we're the only mammalian species that can do a pincer grip. And that's part of the reason why none of the animal models that we use can really adequately model the disease, because no other animal species has a pincer grip. And this is a classic presentation of motor neuron disease. But what it's reflecting is the large representation of the thumb and the index finger of the cortical motor neuron region. And we've been interested then to try and link the two axes, the upper and the lower motor neuron, in some sort of fashion. But we'll see that all of the central diseases, Parkinson's disease, very much associated with gut abnormalities, and perhaps that's the lead into the preclinical phase of the disease. Separately, all the various lower motor neuron presentations, whether it's related to diabetes, a secondary effect of cancer, or cancer treatments, what are they doing throughout the neural axis that will help us to better understand and treat? Now, we've been doing this from a very functional perspective, looking at how the brain goes down an activity, excitotoxicity through the neural channels and into the peripheral nerves. In fact, this was Steve Vucic when he started his PhD, looking at the process of spread throughout the neural axis. But what we really would like to do is incorporate that with functional imaging, not just of the brain, but also can we look at the brain, spinal cord, peripheral nerves in these individuals and try to better understand and track the disease. And ultimately, can this be a marker for treatment outcomes? So I think, it's a fantastic development, I think, for Sydney and across various partnerships in the local health districts. And I think there's a lot of potential overlap. And I'm only focusing more on the neurodegenerative conditions, but I think across all conditions of the brain and mind, there'll be unique insights that we can get from total body PET. And in fact, this is thanks to Charcot. Hopefully that by getting a better understanding of the disease, we can come up with more focused treatments and ultimately better outcomes for patients. Thank you.